This is followed by his postdoctoral work at KU Leuven and Harvard Medical School. These are some of the best centers for advanced research. And subsequently, in 2008, he was appointed as assistant professor and associate professor at Tokyo Institute of Technology, another great center, in 2012. And later on in 2019, he assumed the position of professor, followed by his associate professor in 2012 at KAUST in Saudi Arabia. So if I may talk a little bit about his research interest is basically focused on the development of the materials and tools. Tools essentially means the microscopy system, optics, optoelectronics for advancing fluorescence microscopy imaging. This is one of the main interests. Other interests include development of microfluidic-based advanced imaging devices and development of near infrared and short wavelength fluorescent probes. That is basically the I would say the engine of development in as far as single molecule imaging is concerned. Also, he's expert in single molecule tracking. To talk a bit about his publications, he's, as I said, one of the top researchers publishing in all the big journals. Science Advances, Communications Biology, and many more journals of this stature. And we're really fortunate to have him. So I'll just hand over to him so that he can deliver the talk, and we're looking forward to it. So Professor Habuchi, please, take over.
Thank you very much, Arthur, for the kind introduction. It's my great pleasure to be here and then share my research with all of you today. So let me share my screen. And then, okay. All right. So let's get started. The title of my talk today is Microfluidics-based nanoscopic characterization of hematopoietic stem progenitor cell homing. So as Partha mentioned, my lab has been working on the development of tools and materials for fluorescence microscopy or fluorescence molecular imaging. As you know, fluorescence microscopy techniques conventionally is used for visualizing biological specimen structures in the size range between sub-micrometer to, let's say, millimeter. But with the development of new techniques, tools, we are now able to apply fluorescence microscopy to much wider range of specimen with size or dimension between nanometer to centimeter. So my lab, so from the technology development perspective, the nanoscopic imaging and also imaging at the tissue organ or organism level are the frontier of the fluorescence imaging field. So my lab has also worked on the development of tools and material for extending the applicability of fluorescence microscopy techniques. So we have mainly worked on 3 projects. The first project is the development of microfluidics-based advanced fluorescence imaging platform and their applications to cellular interactions under the external shear force. And this is the project I'm going to present in today's talk. We are also working on the development of near-infrared short-wave infrared emitting fluorescent nanoparticles and their application in tissue imaging. In particular, we are interested in plant tissue imaging at the single particle level using these new tools. And then we are also working on the development of new single molecule tracking techniques. So as I said, today my talk will focus on the first project, development of microfluidics-based advanced fluorescence imaging platform. So scientifically, the aim or objective of this project is to understand the molecular mechanism of hematopoietic stem progenitor cell homing. So this schematic illustration shows what happened to the blood stem cell when they're in the bloodstream. So hematopoietic stem progenitor cells, when you inject these stem cells into the bloodstream, they're spontaneously transported to bone marrow as final resident. And there, the hematopoietic stem progenitor cell can generate a lot of healthy blood cells. Therefore, this biologically important phenomena has been used extensively to treat some blood disorders, such as leukemia, through the bone marrow transplantation. So let me tell you a little bit about this biological phenomena. First, the hematopoietic stem progenitor cell interact with endothelial cells through the binding of selectin proteins expressed on endothelial to their ligand on the blood stem cell. So this causes tethering and rolling because of the blood flow rolling of the blood stem cell on endothelial layer. And the second step is the activation of integrin on the blood stem cell. And the third step is the firm adhesion and arrest of the stem cell due to the binding of integrin with icons on endothelial layer. And the last step is the transmigration of the blood stem cell into bone marrow. This is induced by chemokine. So there have been a lot of works reported the mechanism of hematopoietic stem progenitor cell homing, and therefore we already know important key players that are involved in this biologically important phenomena, such as initial interaction, cellular interaction mediated by selectin and the ligand, and firm adhesion mediated by integrin and the receptor, and the chemokine-induced firm adhesion, transmigration. So why we need to study more on this topic? The answer is that we still don't know almost anything about the spatial temporal behavior of these key players during the homing of the hematopoietic stem progenitor cell. So it is very important, I will show you later, that it is very important to understand the interaction between these key players in the context of space and time in order to fully understand how the homing or cell migration occurs. So that's why we are interested in developing new fluorescent microscopy platform that allows us to characterize spatial temporal dynamics of selectin ligand interactions at the molecular level during the homing processes. There are a few specific questions that we are interested in addressing through this project. First one is the clustering of ligand receptors. So what is the nanoscale architecture of selectin ligands on the cell surface? So some studies suggested that spatial clustering of the adhesion molecules are important in the efficient cellular interactions. The second question is force-dependent ligand-receptor interactions. How does the nanoscale architecture of selectin ligands affect the selectin ligand interactions and the cell rolling under the presence of external force? Because the cellular interaction or ligand-receptor interaction occurs in the presence of external force induced by the blood flow, the force may play a very important role here. The third point is the membrane tethers and slings. So how does the shear force affect the morphology of the cell, selectin ligand interactions, and the cell rolling? So recent studies more and more reveal the critical importance of the cellular morphology, nanoscopic cellular morphology on the efficient rolling. So that's the third question we want to address. And in order to address these questions, we've developed several microfluidic-based in vitro cell rolling assays. So let me briefly explain to you how they work. So we use microfluidic chamber and we coated the surface of chamber with recombinant selectin proteins that trigger the initial step of the homing through selectin ligand interactions. Then we injected hematopoietic stem progenitor cell or their model system called KG1A cell into the fluidic chamber using syringe pump, and which mimic the cellular interaction occurring in the bloodstream. Then we applied advanced fluorescence microscopy techniques, including super resolution microscopy or single molecule fluorescence imaging techniques to capture basically the bottom of the cell where that selectin ligand interaction occurs, and then characterize their spatial and temporal behavior. So we tested if our in vitro system can mimic the in vivo cellular interactions. We look at the rolling behavior of an injected KG1. So these small dots are all KG1 cells on the selectin coded surface. So we characterize the number of bound cells and also rolling velocity of these bound cells on the surface against the different applied shear stresses. I'm not going to tell you detail of this experimental results, but basically these experiments confirm that we can, in principle, mimic the cellular interaction or selectin ligand interaction or rolling of the cell occurring in vivo using our in vitro experimental setup. So using this system, we first try to characterize the nanoscopic spatial temporal dynamics of adhesion molecules. So as I said, the initial step of the homing is mediated by the interaction between selectin protein on endothelial cell and their ligands expressed on the migrating cell, such as PSGL1, CD44, ESL1, etc. So we first visualize their spatial distribution on the migrating cell, in this case KG1A cell, and see if there's any nanoscale clustering behavior of their ligand. So these are the immunofluorescence microscopy images of PSGL1 and CD44 captured by standard confocal microscope. We already see some sort of spatial heterogeneity of the spatial distribution of the ligand, but it's not very, very clear. So we switched to super resolution imaging. And then now you can clearly see that both PSGL1 and CD44 form nanoscale clusters, but they form different types of clusters. Much smaller clusters were observed for PSGL1, whereas much larger patchy shaped clusters were observed for CD44. So we first focus on one of the selectin ligands, CD44, and then see what happened to this ligand during the initial step of the homing, which is the tethering and rolling of the cell. So we captured a super resolution fluorescence images of CD44 on KG1 cell before cell roll on selectin surface and after it rolled over selectin surface. As I showed you in the previous slide, before the rolling, we observed patchy clusters about 200, 250 nanometer width, and which indicate that CD44 molecules distribute on a microvilli of KG1 cell, which are the protruding structure existing on the cell. After the rolling, we found that the spatial organization of CD44 changed completely from patchy to elongated network-like structures, and color bar shows the Z position of CD44. As you can see, most of the elongated clusters of CD44 are located at the bottom of the sample. Therefore, this part is bound to surface deposited selectin. So from this experiment, we learned that shear stress exerted on the cell due to the binding of the E-selectin and CD44 is essential for the reorganization of CD44 during the cell rolling. So now the next question is this clustering important for the rolling or not? So we treated KG1 cell with cyclodextrin, which basically disrupt the lipid raft microdomains existing on the cell surface. By disrupting lipid raft, we found that most of the CD44 clusters existing on the cell disappeared. We still see some. And then we look at the CD44 patterns on the treated cell after the cell rolling. And we found that there is almost no change in the spatial clustering behavior of CD44 before and after the rolling. So the clustering of CD44 is essential for the nanoscale reorganization of CD44 during the cell rolling on E-selectins. So now we want to know if such a nanoscopic change and the spatial organization of selectin ligands affect the macroscopic cellular interaction, more specifically, cell rolling behavior of KG1 cell on the selectin surface. So these are the time-lapse bright field optical microscopy images of control cell and lipid raft disrupted cells. We found that the velocity of the rolling increased significantly when the lipid raft was disrupted. Also, we found that the number of bound cells decreased gradually over time for the lipid raft disrupted cell. Control cell binds the surface more strongly. So these findings suggested that the clustering of CD44 promotes slower rolling of the KG1 cell, and also the nanoscale reorganization of CD44 contributes to the stable rolling of the cell over E-selectin. So from these experimental observations, we proposed the following model for the initial step of the hematopoietic stem progenitor cell homing. In the controlled cell, you have a clustering of selectin ligands, CD44, and this clustering causes stronger binding between E-selectin and its ligand, and thus slow rolling. And the external force is persistently exerted under this condition to the cell due to the strong binding, which causes the reorganization of CD44 clusters from patchy to elongated shape. And this reorganization of CD44 cluster further strengthened the binding and cause stable rolling of the cell on the selectin surface or endothelial cell. On the other hand, when the lipid raft was disrupted, because of the disruption of the CD44 clusters, we observe faster rolling. And this is because of the weaker binding of selectin and ligands in the absence of cluster. And the weaker binding may not be able to exert force to the cell enough time to reorganize CD44 and actin cytoskeleton. And therefore, the cell shows unstable rolling and then detach from the surface quickly. So what we learned from this part of the project is that initial step of hematopoietic stem progenitor cell homing is far more dynamic than previously thought. So previously, initial step was thought to be described by a simple selectin ligands binding and dissociation, but our study suggested that this step is regulated by highly complex spatial-temporal interaction between the cell and E-selectin. So that's what we've learned at very early phase of this project. And also this result gave us a direction for the next step. So far, we look at the spatial distribution of selectin ligand using a fixed cell. So now we want to characterize their behavior in real time and possibly at the molecular level. So that in the next part, I'll talk about a single molecule characterization of space-temporal dynamics of adhesion molecules. So let me begin this part by explaining two movies I showed in this slide. So left side slide shows the time-lapse fluorescence images of KG1 cell rolling over selectin captured in real time. So you see the migration or rolling of the cell, and you also observe the formation of a weird protruding flexible cellular structure both on the back and front side of the cell. And these structures are called membrane tethers and membrane slings, and actually they are part of cell membrane. When you look at the similar real-time live cell fluorescence microscope images of the lipid raft disrupted cells, you first notice the fast rolling velocity of the cell, and also you notice that the formation of membrane tethers and slings is extremely inefficient. And we already know that CD44 clusters in the control cell, but is disrupted in the lipid raft disrupted cell. We also captured scanning electron microscope images of control and treated cell and found that control cells are coated by microfluidic sigin protruding structure are absent in lipid raft disrupted cell. So these observations suggest that spatial clustering of CD44 plays an important role in both the initial tether formation that was demonstrated in the previous part, and also the elongation of the tethers against the tension exerted to the tethering point. And second, the formation of the membrane tethers and slings enable slow and stable cell rolling. So the formation of such transiently appeared protruding structures are quite important for the stable cell rolling. And it is likely that microvilli plays a key role here. So we slightly modified experimental system to capture the real-time live cell single molecular behavior of the rolling cell. We here inserted objective piezo and then scan the image along z-axis with 0.5 or 1 micrometer step to capture images at different z positions during the cell rolling experiment. Since the rolling of the cell is occurred in the time scale of seconds to minute, we can scan the entire z-axis range of the cell in several hundred of milliseconds. With that, basically we can capture 3D structures of the rolling cell. So for sure, when you look at this XY and XZ image of the rolling cell, the fluorescence image obtained from the cell body is distorted because of the fluorescence signal from above or below the focusing plane. But when you look at the tethers or slings, you can nicely capture both XY and XZ image of this temporary form protruding structures, because their width is such very narrow. Therefore, even with standard wide field microscopic configuration, we can nicely capture such structures. And eventually, you can see individual tether and slings with some bright spot at the end, which suggests the clustering of, in this case, CD44. Now, we look at another selectin ligand, PSGL1, and then characterize their interaction with E-selectin in this part. Again, let me play this movie. Hmm. Okay. Yep. So this video shows the real-time fluorescence images of KG1 cell rolling on E-selectin. And now we look at fluorescence from PSGL1 ligand on the cell. You can clearly see the formation of membrane tethers and slings, the back and front side of the cell. And when you look at the rolling cell on tether and sling very carefully, you notice that you can see distinct spots along these structures. And actually, these spots are individual PSGL1 molecule existing along the tethers and slings. We confirms that by comparing the fluorescence intensity obtained from individual spot and individual PSGL1 amino labeled PSGL1 molecules. Then we determine the number of PSGL1 at the tethering point along the along the tethers and at the anchoring point. Anchoring point is between the tethering point and the cell body. As I said, the PSGL1 molecules existing along the tethering slings are single molecules. On the other hand, the average number of PSGL1 molecule at the tethering point is about 4 molecules, and anchoring point 2 molecules. So this result confirmed again that the clustering of selectin ligands is quite important for the formation of or binding of selectin ligand and actually formation of the tether and slings. Since we can count the number or distributions of individual PSGL molecules, we can actually determine the number of PSGL1 molecule per tether or per slings. So we first determine the lengths of the tether and sling at different shear stresses. So from 10 micron to 30 micrometer, depending on the applied shear stress. Then we determine the distance between the adjacent PSGL1 molecules. And with these two data, we can determine the total number of PSGL1 per tether and slings. And interestingly, the total number is almost independent of the shear stress. So this observation allow us to propose this model for the selectin ligand binding. So CD44 PSGL1 are distributed on microvilli protruding structure on the cell, and their cluster binds to E-selectin on the surface, and then due to the external shear stress, you form the tethers. But this tether is formed from the single microvilli because the shear stress doesn't affect or length of the tether and number of the PSGL1 are not related. So you always have a constant number of PSGL1. Therefore, it is strongly suggested that single microvilli extend and form membrane tethers. And when this happens, selectin ligands, in this case PSGL1s, are localized on the tether and slings. That increases the probability of the binding of selectin and their ligand during the rolling on tether or slings. So that's why formation of tether and membrane slings help a lot stable rolling of the cell on the surface. We also found that these selectin ligands shows diffusional motion along tethers and slings. So we determine the speed of the motion and mode of the motion of these molecules. This figure shows the distribution of diffusion coefficient observed on the rolled cell and control cell. You clearly see that on the rolled cell, on tether and slings, PSL molecule shows much faster diffusion. The right side panels shows the mean square displacement versus time lag plot. You clearly see the linear relationship between time lag and mean square displacement for the PSL1 on tethers and slings. So that means PSL molecules on tether and slings shows random 1D diffusion. On the other hand, mean square displacement versus time lag plot obtained from the control set clearly shows the subdiffusion, which is analyzed by confined diffusion model. So this result suggests that the mode of the diffusion is also significantly affected by the formation of tethers. We also observed the merge of multiple tethers during the cell rolling. So this is a real-time video. So the merging of multiple membrane tethers occurred in a time scale of, let's say, second. So it's a quite fast process. So it is known that microvilli has a cytoskeleton backbone in the control cell. So we experimentally determine if actin cytoskeleton still exists in the membrane tether and slings. In the control cell, this two-color super-resolution imaging clearly shows that CD44 shown in cyan and actin cytoskeleton shown in yellow are spatially overlapping. On the other hand, when you look at fluorescence images of CD44 and actin on the rolling cell, you see the formation of membrane tether or sling, but you only observed a small fragment of actin along these structures. So basically, during the formation of membrane tethers, actin cytoskeleton detached from the cell membrane. So there is no actin cytoskeleton in the cell membrane. Result of this is the faster motion of selectin ligands because they are no longer anchored to actin cytoskeleton, and also shows the random motion. That was what single molecule tracking experiment suggested. So according to this model, formation membrane tether and slings play a significant effect in the efficient binding of selectin ligands to the surface selectin due to the fast diffusion on tethers and slings when it's formed. So together, our single molecule live cell imaging experiment suggested that the migrating cell has a very smart mechanism to utilize a limited number of selectin ligands for the efficient selectin ligand binding, therefore the efficient tethering rolling of the cell in the first step of a hematopoietic stem progenitor cell homing. And this is done by the formation of membrane tethers from single microvilli that enable the localization of selectin ligands on this protruding structure, and the detachment of the actin cytoskeleton from the membrane during the membrane tether formation allow for the rapid motion, therefore the efficient binding. So these are the findings that we observe from live cell single molecule level characterizations. So we more or less understood what happened at the initial step of the homing. The next question is that what would be the effect of the endothelial cells or endothelial layer in the in vivo cellular interactions? So the actual hematopoietic stem progenitor cell homing occurred in the blood vessel. So that means migrating cell interact with endothelial cells. So it's not only about selecting ligands, but other cellular structures or function may affect the initial step of the homing. So this is what we tried to characterize in the next step. So we developed a modified experimental platform for addressing this issue. Instead of selecting, recombinant selecting on the surface of a Freydig chamber, we cultured Chinese hamster oval cell, CHO cell, which is transfected with E-selectin. So that means CHO cell form a monolayer on the surface of Freydig chamber surface, and then express E-selectin ligands on their surface. So now we can also take into account the effect of cellular structure and architecture in the initial step of the homing. And here we characterize the migrating cell not only by using fluorescence microscopy, but also using scanning electron microscopy. We first confirm that CHO cell cultured in the microfluidic chamber express E-selectin, and then they are healthy during the cell rolling experiment. So panel A, it shows the 0-hour and 40-hour incubation of CHO cell in the fluidic chamber. As you can see, after 48-hour cell culturing, the surface coverage percentage is more than 90%. So most of the chamber surface was covered by monolayer of CHO E cell. We also determined the viability of the CHO E cell before and after the rolling experiment, and there's negligible change in the viability. We also checked the expression of E-selectin on CHO E cell. So this is bright field microscopic image, which capture the morphology of the cell. And when you look at the fluorescence image obtained from the same area, you can clearly see the fluorescence from the CHO E cell. So we also noticed that the expression level of E-selectin depends on each cell. So this video shows the rolling behavior of KG1 cell on underlying CHO E cell. So you can see that a lot of KG1 cell actually shows a rolling behavior on CHO E cell. And we also confirmed that this cellular interaction is caused by a specific selectin ligands interaction. So we first look at number of bound cells on CHO E cell and negative control CHO cell, which doesn't express E-selectin. So we clearly observe a large difference. And we also tested the number of bound cell in the presence of EDTA. So selective ligand interaction require calcium. So when you chelate calcium using EDTA, there's almost no binding. So we confirmed that the rolling-like behavior we observed in this experimental system is caused by the specific selecting ligand interactions. And we also tested the rolling velocity of the migrating cell at different shear stresses. We observed slightly different shear stress dependent rolling velocity compared with the E-selectin surface coated by recombinant E-selectin. So we already observed some difference between recombinant E-selectin and E-selectin expressed on the cell. And again, we applied 3D imaging of the migrating or rolling KG1 cell And this panel shows XY view, basically top view, and the top panel shows the XZ view. So you can nicely see the formation of tethers and then slings. And then we can characterize the behavior of individual selectin ligands that are rolling over CHO-E cell. We also tried to preserve the transiently appeared membrane tether and sling structures. So these top images are obtained for live sample. The first images clearly shows the formation of tether and slings. And when you look at the bright field image obtained at the same position, the white dot shows the location of the tethering point. And clearly you can see that tethering point exists on the underlying CHO-E cell. That also confirms that the formation of tether and slings are caused by the selectin ligand interactions. We spend a lot of time optimizing the protocol of fixing rolling KG1SL without damaging the very fragile membrane tether and sling structures. After a lot of optimizations, finally, we were able to preserve these fragile structures. And now you can see tether and slings preserved in the fixed cell. And again, you can see that tethering points are located on the surface of underlying CHO-E cells. So this optimization of the fixing protocol enable us to capture electron microscope image of the migrating KG1SL. So this image shows the scanning electron microscope image of KG1SL rolled over CHO-E monolayer. So flat cells are CHO-E cells, and you can see that surface of the fluidic chamber is nicely coated by CHO-E monolayer, and round shaped cell is KG1SL. And when you look at the KG1SL in the center, you can clearly see the formation of tethers or slings, so protruding structures extending from cell surface, and they attach to the underlying CHO-E monolayers. We also look at the scanning electron microscope scope image of KG1SL rolled over recombinant E-selectin coated surface. We found the formation of tether-like structures, but at the same time, we also observed the significant elongation of the cells. So these scanning electron microscopy experiment clearly suggests that the cellular structures is quite important in or play an important role in the rolling behavior of the hematopoietic stem progenitor cell. We, at this stage, we still don't know how exactly the cellular architecture allow to maintain spherical shape of the rolling cell. It is likely that the elasticity of the CHO-E cell or endothelial layer allow the rolling cell to maintain its integrity. This is something we can learn using the experimental platform we developed in this part of the study. So all these results that I showed you indicated that cellular architecture play an important role in the tethering and rolling of the cell and the hematopoietic stem progenitor cell, and also the spatial clustering distributions of selectin ligands are important for the stable rolling. So we already know these are important, but so far we were not able to visualize, characterize these two factors simultaneously. So we are currently trying to build a new experimental platform that can characterize both cell surface morphology and localization of adhesion molecules during the cell rolling. So this slide shows the basic scheme of this new experimental platform. So we use both 3D STORM, as 3D super resolution fluorescence microscopy, and total internal refraction, or TIRF microscopy, to simultaneously capture the surface architecture of the cell and the location of the selected ligands. So TIRF microscopy basically capture the surface morphology of the cell, and then super resolution microscopy allows us to localize locations of selected ligands in 3D space. And then we merge them and then kind of build the image shown in this schematic illustration. So that's what we wanted to do here. So as you probably know, TIRF microscopy can capture fluorescence only from the surface or very close to the surface, because evanescent field generated by the total internal refraction of the excitation light doesn't propagate through space. It's localized close to the surface. And actually, the field intensity decreased exponentially from the surface. So basically, you can convert the fluorescence intensity obtained in TIRF microscope experiment to the distance from the surface. When the sample is closer to the surface, you expect to see brighter fluorescence. When the sample is far away from the surface, you see weaker fluorescence. And of course, you don't have a propagating light, so the depth, maximum depth, is only several hundred nanometers in this case. So we applied this to determine the 3D morphology of the cell surface. So we use membrane stain called membrite, and then stain the KG1S cell almost homogeneously. So it doesn't matter where we look at the cell surface, the density of the stains are more or less the same. Therefore, intensity directly reflect the distance from the surface. So in the right, I show you the raw intensity image we obtained in the TIRF microscopy condition. Then we process the image using a suitable filter to highlight the cell bottom, mainly microvilli or elongated microvilli or even tether and slings. And you can clearly see that some parts are much brighter than others. And there are also regions with very dark fluorescence. So from this intensity map, we generated the surface 3D map of the cell bottom. Top image shows the Z profile on the bottom of the surface in the range of 120 nanometer from the bottom. The bottom, you have a top view of the bottom of the cell. The color bar shows the Z positions. Larger value means it's closer to the tip of the cell or tip of the microvilli, and the smaller value means they're close to the cell body. So you can nicely generate 3D surface map. Then we captured 3D super resolution images of selected ligands, or in this case CD44, and then overlay two images. So this is the projected image. This is a 3D view. So with these red or blue dots are the localization determined by the 3D super resolution imaging. And then this 3D profile surface map shows the surface morphology of the cell. So we did some quantitative analysis to evaluate if we can nicely localize the selecting ligands on the surface map generated by turf microscopy. So we, in the middle, it shows the normalized turf microscopy surface map obtained from one of the microvilli captured in the experiment. Right side, it shows the normalized fitted surface of a single molecule. So you see a large number of localizations obtained from the CD44 super resolution measurement. And then we generate a surface map using this 3D coordinate of these spots. And we obtained this image. So clearly, there is a nice similarity between the topographic map obtained from turf microscopy and 3D super resolution microscopy. So now we can say that we can overlay a surface, cell surface map and 3D coordinates of individual molecules. We look at the control cell and rolled cell. So in this experiment, we use recombinant E-selectin coated surface. So you clearly see patchy clusters of CD44 in the control cell and more elongated network-like clusters after the rolling. Then we overlay its cell surface map with the 3D coordinates of CD44. And then we obtain this z-axis profile of CD44 and PSGL1. Oops. Right side data was obtained from control sample, and left side data was obtained from rolled cell. Z-axis correspond to the Z positions, and each dot shows the locations of individual CD44 molecules. When you look at the control, you see more evenly distributed CD44 or PSGL1 along Z-axis. When you look at the rolled cell, you clearly see that majority of the ligands are located closer to the tip of microvilli. So this behavior can be also seen in this Z position versus density map. Control cell shows more uniform distribution along Z-axis, but the rolled cell clearly shows a larger distribution or density closer to the tip of microvilli. We also compare the microvilli and tethering points. When you look at microvilli after rolling, CD44 and PSGL1 slightly shows the larger distribution toward the tip of the microvilli. But when we look at the tethering point, it is more clear that most of the CD44 PSGL molecules are localized very close to the tip. So therefore, this result also demonstrated that tethering at the tethering point, we have larger clusters of selecting ligands, and such a clustering played an important role in the stable and efficient binding of cell on the surface. And we are currently trying to characterize the locations of selecting ligands on individual microvilli so that we can visually capture their spatial location with respect to the cell surface. And this is something what we are trying to do now. Okay, so let me briefly tell you another project we recently started. So we are for sure interested in the initial phase of this hematopoietic stem progenitor cell homing, which is the tethering and rolling. But we are also interested in the later mechanism, the later stage of the homing. So we are particularly interested in this transmigration step induced by chemokine. So now we are developing new experimental platform that enables to capture and characterize this step. So idea is that we use fluidic chamber, and we have a two-part, bottom part and top part. Bottom part is filled by gel matrix, and we have a monolayer of endothelial cells. And then top layer, we have migrating cells. And then we added chemokine in the bottom part that should trigger the transmigration of the rolling cell or transmigration of the homing cell. And then we should be able to capture and characterize their behavior using advanced process microscopy. So that's what we recently started project. Okay, so let me summarize my today's talk. So in this project, we developed new microfluidics-based experimental platforms that enable the direct capture of nanoscopic spatial temporal dynamics of adhesion molecules, for example, selecting ligands interaction at the molecular level. One is the super resolution imaging of spatial temporal reorganization of the adhesion molecule. And second, single molecule live cell fluorescence imaging of selecting ligands. And third, electron microscopy and turf microscopy based characterization of cell surface morphology change. So I want to thank people contributing to this work. So this is the lab photo that we took quite recently. I want to thank all the members of current lab. I also want to thank previous lab members who contributed to this work. And also I want to thank my collaborator, Professor Jasmine Mirzaban, and people in their lab who contributed to this project. I also want to thank financial support from our university council. And lastly, I would like to thank you very much for your kind attention, and I'm happy to take your question if you have any. Thank you very much.
Thank you, Professor Abuchi. It was a very interesting talk and very absorbing. What we witnessed basically now is the future of biology and the ways to understand biological problems using physics, chemistry, and engineering tools. And this really shows how the biology should be pursued using advanced optical tools and techniques. Just want to inform that the talk was YouTube live and will be available on YouTube channel, for present and budding researchers. So this will be a great contribution in that direction. So I would like, if anyone has any question, they can just go ahead and ask. That would be a great discussion. So any questions that some of you may have? Take that. Yeah, you can. No, you take that. No? Okay, just take that. Okay. I want to ask you something. I mean, before anybody goes in, just for my understanding, how do you differentiate between binding based on slowdown based on binding and slowdown due to proximity to the surface?
Yeah, so as you may know, we have laminar flow in the fluidic chamber. So we have a parabolic flow profile
Okay.
in the fluidic chamber. So when the cell get closer to the surface, the velocity become slower. But still, the velocity of the cell flowing freely close to the cell surface and the cell that actually interact with the surface and then rolling behavior has a very different, let's say, velocity. So at least an order of magnitude different velocity is expected, and that's what we observed. So the distinguishing between free flow and then rolling cell is not that difficult in our experimental setting. Right, so we track the cells. So if the cell shows the mean velocity of, for example, 100 micrometer per second, it clearly shows it's a free flowing. If the velocity of the cell after tracking give us an average velocity of 5-10 micron per second, then that reflects the cellular interaction.
So you're able to differentiate between these two kinds. I mean, the velocity slowdown due to both these reasons.
So your question is distinguishing between
These two slowdowns. So you're able to
yeah,
do that,
yeah.
right?
So I didn't talk about how we actually characterize rolling behavior using microscope images. So we track individual cells in the field of view in each frame, and then generate trajectory of the motion or flow. And then we calculate the time average velocity of the cell that are in the field of view. And each cell basically give us time average velocity. And when you look at the distribution of time average velocity, it's rather obvious. There are two different populations, bound one and then free flow one.
Okay. Right. Anyone else want to ask any question? Yeah. Neeraj, go ahead. Yeah.
Hi, sir. I have actually one doubt. Sir, you can mute. Hello?
Yeah, go ahead.
Yeah, so I have one doubt from that flow cytometry system, the cytometry system. Actually, the thing is that there you presented three different systems. So I want to ask that, is it because we are targeting three different organelles of the same cell, or is it because of some different reason?
Oh, okay. So your question is why we've been developing different types of experimental platform?
Yeah, three different lasers here.
Yeah. Okay. So we are driven by scientific questions. So no, if we want to know the basic molecular mechanism of the initial step of the homing, the experimental platform we developed first was sufficient. But then we asked the question, what would be the contribution of cellular architecture on the homing behavior? Then we had to develop slightly different experimental platform by culturing one layer of cell in the platform on the fluidic chamber. And then we also wanted to ask if how exactly the morphology change of the cell affect the localization of the selecting ligands, and how does that affect the first step of the homing. And that's why we decided to develop a turf microscopy combined with 3D super resolution microscopy platform to actually characterize and analyze the behavior. And so basically, we always have a scientific motivation to develop different types of experimental platform. Does that answer to your question?
Yeah, thank you, sir.
Right. Anyone else who has some questions? Online, do you see, Arvind, anyone who want to ask question? Okay, just a minute. I'll just look at it. So Arvind want to ask how to take care of motion blurriness of single molecules.
Okay, so as I told you briefly in the presentation, the time scale of cell migration, cell rolling on the surface is it's second to minute. And when we look at the motion of individual molecule, we are interested in the motion of a molecule occurring at much faster time scale. So for sure, when we look at the motion of the molecule occurring at the time scale of seconds to minute, this is affected by the motion of the cell, but we are interested in we wanted to characterize the speed and mode of the motion occurring in the time scale of sub-second. So for this time scale, we don't have to worry too much about the motion of the cell or motion of the tether sling structures. They don't affect our analysis that much.
Okay. Yeah, we'll just see if any more questions are there. Anyone else who want to ask? Okay. Yeah, I don't see any more questions, but really a great depth that we have gone through, the type of system that you developed, and that actually is going to help biology in a big way. I see that, I mean, this is going to will be replicated by many groups to to learn or understand biology in future. So once again, I mean, I would like to just thank you for making time for this and really delivering a great talk. So we can just, yeah. Yeah.
Thank you so
Okay.
much.
Thanks, Professor Havuchi. Thanks a lot.
Thank you so much, and have a good day.
Thank you. Thank you. Bye.